Okay, so let's now go to other proteins. But before we do that, let's dispel some of the uh, common misconceptions that blue, deoxygenated blood is blue and oxygenated blood is red. Okay? You may see it because our veins, okay, if you look to our skin, appears greenish or bluish, and we think that deoxygenated blood is blue. Okay? So here are flasks of deoxygenated blood on the left. Okay, so it's blood sample filled with carbon dioxide sample. So you see it's like purplish in color, and then the oxygenated blood appears red. So, you know. So let's examine keratin. Okay, so touch your fingernails and your hair. Compare the texture. Okay, you may not believe it, but they're composed of the same proteins, keratin. Okay, so let's look at the structure. I don't have the primary structure of keratin, but there's the secondary structure, predominantly alpha helical. Okay, and then there's a alpha helical domain and there's a globular domain. This is the monomer. They can form dimers. No? So the alpha helices wind around each other to form a coil, if you will. So your monomer becomes a dimer, and then they line up with each other to form the protofilament. And then more of these lining up together is a protofibril, and then eventually it becomes keratin. Okay, so let's solve the seeming problem as to why hair is soft, whereas fingernail are harder. Yeah? So hair, as you can already read, has fewer crosslinks, so that makes it softer. And in the fingernail, there's lots of crosslinks among these filaments, and that makes it harder. I found this cross-section view of hair. Okay? So you have the keratin, okay, running across the direction of the strand of hair. So as mentioned, you have the alpha helix of the keratin forming the coil, and then the protofilament, protofibril, and then the intermediate filament, and then the cells. Okay, since we're talking about hair, let's talk about some of the things that some of you do with your hair, and that is have a perm or have your hair rebonded. Well, before the pandemic anyway, which is really just a manipulation of the hair structure. So what you have in hair, you have the keratin, and in keratin you may have the cross-linking of disulfide linkages. So what does your beautician do? They just apply a reducing agent to your hair. It breaks apart the disulfide linkages, and then they become reduced to the sulfidyl groups. And then if you're getting a perm, curls the hair into place, lets it sit for a while, okay, and then reoxidize it into a different connectivity. So this is the original connectivity, so your hair may be straight or curly at this stage, and then once you now get the curl, it's oxidized, but it's no longer in the original configuration. Okay, so your hair now becomes curly. It's the same if you're getting a rebond. Instead of curling it up, it straightens it up and then reoxidize it into the straight configuration. So that's it. How about silk fiberwin? Okay, so fiberwin is made by silkworms and spiders. They're really just long regions of anti-parallel beta sheets. Some parts are not in sheets, but again, you have beta sheets. These are multiple repetitions of glycyl alanine, glycyl alanine, glycyl serine, glycyl alanine, glycine, and so on. Okay, and in this repeating structures, okay, they form beta sheets. And the thing about beta sheets is that the side chains are parallel to the main direction of the sheets. So the beta sheets can like stack in front of on top of each other. And once again, here's the secret. Okay, so you have of these repeating units: glycine, alanine, and so on. So glycine, you just have hydrogen, so it's a small steric requirement. And then alanine, well, relatively small, but you cannot replace it with something that's larger because they cannot stack on top of each other anymore. So they sort of like yeah, they stack on top of each other, giving it strength. So here's a ball and stick model picture with the planes of the peptide being shown, the beta sheets being shown. Here's a side view. Okay, so you have like this is one layer of peptide, another layer, and so on. So it's also color coded. So it's glycine, so they fit very nicely. And then the alanine or the serine fitting also very nicely on top of each other. So you have this very strong sheets. Okay, but how about the spider web? It's the same. No, it's still a combination of the beta sheets, the alpha helices, and some random coils. So this is a very good combination because you can have strength due to the beta sheets, and then you can have the flexibility due to the alpha helices. Okay, so note that again, one of the problems with the illustrations is that they tend to be static. So these are coils; these are just held by hydrogen bonds. So you can imagine these coils to be more flexible. No? So they can just like a spring. No? You can move them back and forth. Okay, so that part imparts flexibility, this part imparts strength, such that okay, if you've ever been what call it sprayed with spider web, okay, if you go to your garden and there's a spider web along the way, you notice that it's annoying. And it's strong. It's actually stronger than steel on a per unit mass basis. No? So you think, it's a spider web. It's easy to break, but consider the mass involved. It's just much stronger than steel. Okay, so other interesting molecules, kinesin. Okay, so at least that's how I pronounce it. I just found this from the net, downloaded it. So kinesin is a motor protein. Okay, that carries a large cargo. So it carries whatever this is in a microtubule. So hydrolyzing one adenosine triphosphate. So if you recall your high school chemistry, high school biology, ATP is considered the the energy currency of the cell. So, no? So this is how things could move within the cell. Okay. How about actin and myosin? Okay, so flex your muscles. Okay, so what happens when you do that? The myosin and the actin and the myosin muscle fibers just move past each other to either relax or to flex now. So again, these are just protein movements in your muscles. Okay, so some other trivia. The largest protein is titin. Okay, so also known as connectin. It's okay. I'll leave it to biology, anatomy, or whatever it is. Flexible intrasarcomeric filamentous protein. Okay, so I got this in quotation because again, just quoted this from somewhere. That functions as a molecular spring, which is responsible for the passive elasticity of muscle. Okay, so you owe it to your titin. So it's large. Now the human variant has about 34,350 amino acids, and the molar mass is 3,816,188.3 Daltons. Okay. So if you're not familiar with Dalton, this is just equivalent to grams per mole. Or in other words, if you have 1 mole of titin, it will weigh 3.8 something grams. 3.8 million something grams. Amazing, right? For the mouse homolog is actually even larger, made up of 35,213 amino acids. So molar mass of almost 4 million that one. So 4 million grams per mole. Wow. Can you imagine? So that makes it also the longest. Another trivia that now makes it the longest word, okay, in the English language. Okay, so 189,819 letters, and it says they report that it will take you 3.5 hours to pronounce correctly. If you make a mistake, it's not over. Can you imagine? So this is the start of the word. Okay, so time to make that concept map and answer the Moodle objectives.